Hello, my name is Miriam Alhamdin, and today I'll be introducing you guys to RNAi pathway. So in eukaryotic cells, RNAi pathway is an important cellular mechanism that plays an essential role in cell immunity and the regulation of protein synthesis. Depending on the process, the RNA pathway can produce small interfering RNA or microRNAs. In this presentation, I'll be focusing on small interfering RNAs, which relies on double-stranded RNA. The RNAi process is triggered by double-stranded RNA. Double-stranded RNA in the cell is preserved as an abnormality, even if it was naturally produced by the cell. The RNAi pathway resolves that abnormality. Double-stranded RNA can arise from several different causes, but it's not fully understood how they can arise in all scenarios. But it is understood that double-stranded RNA can arise when two inverted copies of RNA fragments bind in a sense and antisense orientation. Those copies attract and can form a double-stranded RNA. Furthermore, in plants, RNAi act as an effective antiviral agent, and it is currently used as a powerful tool in understanding viral infections, genetic disorders, and creating treatments for them. But let's take a closer look on how RNAi pathway function in this cell instead. Okay, so first, when a double-stranded RNA is present in the cell, the RNAi is triggered. Second, an enzyme in RNAi called dicer dices the double-stranded RNA into small interfering RNA, also known as siRNA. siRNA are only 21 base pairs, which is too small to be translated into functioning proteins. Third, some of those siRNAs are then utilized by another protein complex called RISC, which is also part of RNAi pathway. Some siRNA binds with RISC, which unwinds the siRNA. Fourth, the unwinded siRNA is used to target the corresponding mRNA using the antisense strand. Fifth, the siRNA binds into the corresponding mRNA in the genome. Sixth, an enzyme from the RISC compound cleaves the bound mRNA, disabling it from being transcribed in that tissue. A great example of this mechanism in action is demonstrated in the color of soybeans. Chalcone synthase is an essential enzyme for the synthesis of anthocyanin pigments, which gives tissues such as flowers and seed coats their red, blue, and purple color. White-colored soybean contains 6 functional chalcone synthase genes that are close by, and 3 that are far away in the genome. Genetics can be a little messy. 3 chalcone synthase genes are in cluster A and 3 in cluster B. Because the 2 clusters are inverted repeats of each other and they're close by, they form a double-stranded RNA when the mRNA is transcribed. Here's where the RNAi pathway starts. The double-stranded RNA gets attacked by dicer, which dices it into siRNA. Since the double-stranded RNA is torn up, it's impossible to translate it into proteins. But remember how genetics can be a little messy? And there are chalcone synthase genes on different chromosomes, and they still get transcribed, but they don't get translated, and that's because of RISC, which uses the unwound siRNA to bind with chalcone synthase 7 and chalcone synthase 8 mRNAs. When the RISC complex cleaves the bound chalcone synthase 7 and chalcone synthase 8 mRNA, it leaves the soybeans with all the RNAs of chalcone synthase being disabled, making colorless soybeans. On the other hand, black soybeans have a deletion in cluster B, which extends to chalcone synthase 4 on cluster A. And since there is a deletion in cluster B, the double-stranded RNA does not form, and a single-stranded RNA is formed, expressing the color, because the RNAi pathway was not triggered. But how was this pathway first discovered? Well, it began with Napoli et al. Research paper experimenting with transgenic petunias to make a darker purple petunias that doesn't naturally occur by overexpressing chalcone synthase gene, as it is an essential enzyme for the synthesis of anthocyanin pigments, which gives flower their red, blue, and purple color. And through running different experiments with transgenic petunia breeding, they ended up with flowers that had patterns that contained white, and there was a large proportion of fully white flowers. And through running different experiments and tests to understand how he ended up with white flowers when he was trying to make dark purple flowers, he was able to conclude that it was due to cosuppression, in which overexpressing the gene caused it to be suppressed, which was a newly discovered phenomenon in transgenic plants in the early 90s. With more research, now we know that Cosuppression is the RNAi mechanism. Currently, using RNAi, scientists have developed crops such as non-browning Arctic apples, nicotine-free tobacco, decaffeinated coffee, and non-allergenic peanuts. Also, RNAi is commonly used in research as a mechanism of silencing genes to understand their function. RNAi has significantly accelerated the rate and pace of biomedical research, and as of 2021, there has been 3 approved treatments for 3 rare hereditary diseases, and there's currently 7 other candidates in progress. The RNAi pathway is also expected to be one day readily utilized therapeutically to treat genetic disorders, cancers, and infectious diseases. And it's probably safe to say that for 30 years since it was first observed, this could be just the beginning. Thanks for watching, and I hope you found this video helpful.
Created using Powtoon.